to do the reading. Some people find that they just like going through the slides and listening and might listen to the recorded ones over again. So whatever works for you, but usually after 3 times, it kind of ingrains itself in your brain. All right, so prokaryotes comes from the Greek words for pre-nucleus. We know eukaryote, our human cells, we have a true nucleus. So that means, it really means true nut for eukaryote. So the nucleus was seen akin to a nut or the seed or the kernel of the cell to the early microscopists. So our prokaryotes are basically going to be our world of bacteria. And our bacteria are all unicellular organisms. There aren't any that are multicellular bacteria. And the world of prokaryotes is divided between, we start looking at the world of living things being divided amongst the 3 kingdoms of archaea, bacteria, and eukarya. And so, you know, a lot of our focus is really specifically going to be on these prokaryotic bacteria. So our prokaryotes, as we went through before, they have not a true nucleus, but the material, the DNA of the prokaryotes is typically in a nucleoid or central region of each prokaryote. They don't have any histones. So unlike our eukaryotic DNA, which will wrap around and organize around the histones to help our DNA compact into the nucleus, but they do have some small proteins that they associate with, those naps that we talked about last time. The bacteria also don't have organelles, so they don't have their own endoplasmic reticulum, Golgi apparatus, any of that stuff, all right? But what we did talk about a little bit last time, and we're going to talk about some more to get it really ingrained in our brains, is we're going to talk about the bacteria peptidoglycan walls. Remember, we don't have peptidoglycan in our cell walls of our eukaryotic cells. So it is really one of the most excellent targets for us to develop antimicrobials, antibiotics, to attack the bacteria without also attacking our own cells. Now, the archaea, which is kind of like called archaea because it's a more ancient or archaic form of bacteria, we won't talk about so much. We pretty typically encounter these more in things like hot springs and that. They really aren't going to be the pathogens that affect us so much. So we're going to gloss over them a little bit more lightly and focus on the bacteria that either live in symbiosis with our body, doing good things for us, or the bacteria that become pathogenic to us, all right? All bacteria is pretty much going to divide by binary fission. So that's going to be the cell's going to replicate its DNA, and then you'll have a cytokinesis or division of the cytoplasmic material. All right, just creating an identical daughter cell. There isn't sexual reproduction in the true sense. There is sometimes a through what we call reproductive pili. One bacteria might send a plasmid over to another bacteria. And as I said last time, we'll get into that a little bit when we get into more some of the genetics later on. And that would be called like a reproductive pili. Some people call it bacterial sex, but it's not quite sexual. All right, so the average size of our bacteria is about 0.2 to 2 micrometers, okay, in dimension, by either 2 to 8 micrometers in length. This is pretty typically on, you know, obviously a rod shaped, a cocci is going to be more like 2 micrometers all around. And someone was asking a little bit about the size because there is a big range. So I dropped these into the slides right before class started, so they might not be on your slides. You can you know, download this if you want, but it's not something I'm going to test you on, so you don't really need to know. But just to say that The largest bacteria we typically deal with in our labs is called Bacillus megaterium or B megaterium. And these bacteria are one of our larger bacteria, and they can be up to about 4 micrometers in length and up to about 3 micrometers in diameter around. The largest bacteria in the world is about 100 to 300 micrometers, and that's this Thiomargarita namibiensis. As you can see, it has Namibia in its specific epithet, and this is a bacteria that lives in the sort of ocean sediment beds off of Namibia. So there can be some pretty big ones of this that I found exist up to about 750 micrometers. Now, if we take a really typical bacteria, one of our pathogenic bacteria of E. coli, you see that this is only about maybe around a micrometer to a micrometer and a half in width, and it can be up to about 6 micrometers in length. So there's, you know, an order of magnitude of more than 100 times bigger from our average bacterium up to our largest bacteria. The mycoplasma, which we'll talk about a little bit more today, are a unique sort of subset of the bacteria. And these are the tiniest bacteria, and you'll see they have a unique cell wall situation, so that keeps them a little bit smaller. And so they're only about 200 to 300 micrometers in their overall size. So they're really the smallest in their volume. Most of our bacteria, you'll see, are what we call monomorphic in shape, or they have one particular shape. E. coli is always a rod. It's always about, you know, the 1 micrometer by about 3 micrometers on the average in length. All right. Most bacteria are pretty consistent that way. We have a few bacteria that we'll call pleomorphic. And pleomorphic means that sometimes it can be a little bit different shaped. And you'll see this when we get to a lot of our little curved bacteria that are kind of comma shaped can sometimes be a little bit more rod shaped. And some things will be like sort of cocci or into Coccobacillus kind of shape. And we typically identify the bacteria that are pleomorphic, so we don't just kind of stick with one specific shape in our mind. But you can see how that would get tricky for identification. All right. So somehow I went back. All right, so we know our bacillus are rod shaped. We know our cocci are rounded shaped. And then we have a variety of spiral shaped bacteria. The Vibrio, which are really only comma shaped. That's like Vibrio cholerae that causes cholera. Then we have Spirillum that kind of twist around. And then our spirochetes are even more twisty. There's a few bacteria that we'll see, but these usually are things that are more like oceanic sort of bacteria that get star and rectangle shape. None of the bacteria that we'll really focus on this semester will take on these unusual shapes. And what gives it its shape is the rigidity of the cell wall, okay? It'll give it its very, very specific shape. And we'll see the arrangement is having to do more with the plane of division. And what I mean by arrangement is whether it's going to be existing as a single bacterium or whether it will, you know, always go in pairs, being like a diplococci or a diplobacillus, or whether it's going to be in chains or in grape-like clusters, etc. All right, so our spirilla, like I said up here first, we have our Vibrio. This is going to be just that comma shaped. They're always going to have this curve into it. The cytoskeleton pulling on the cell wall, which is very rigid around it, is always going to make it comma shaped. And so, I found this new computer is a little bit happy on the trigger, so it jumps on me every now and then. I gotta try to figure out how to make that a little bit less sensitive. So, you know, we tend to recognize easily the species that are comma shaped. Vibrio cholerae is the one that you need to remember for comma shape. Spirilla spirilla, I'm sorry, not Ia, spirilla, are the ones that have this kind of little twisty shape, okay? And these have a pretty rigid structure that keeps it looking a little bit like pasta, right? Some sort of spiral spaghetti, or not. What do you call that spiral spaghetti? Anybody know? The spiral pasta? Any?
Rotini?
Huh?
Rotini?
Is it rotini? That sounds right. Okay. Somebody here, I figure, has to be Italian and grew up with this. I'm trying to avoid pasta because it makes me fat. So you'll see these are kind of like the little thicker ones, and the spiral's not as deeply twisted as when we get into these spirochetes. And these are really kind of spring-likey, flexible, twisty bacteria. And our one that we always remember as a spirochete is the treponema, which causes syphilis, okay, is our sort of marker of that type. Treponema pallidum, all right. So these ones, like I said, these kind of hang out more almost like on oceans along with algae. They'll help give, a lot of them will carry little chromophores in them that give them color. So we see more star-shaped bacteria living in the ocean, and we're not really going to talk about these too much, but I want you to know that they exist, as well as some very oddly rectangular shaped bacteria. All right, so again, the arrangement is going to have to do with the plane of division. So what you'll see, and this is where we get in deficits with being at home, is we kind of draw it out typically on the chalkboard of when you have your one cocci and it's going to undergo binary fission and split in two, and you get your cleavage furrow down the middle, and it kind of goes from like this, and then they separate, okay? And then they'll kind of stick together and travel as diplococci. When we start getting into streptococci chains like that, each of these diplococci will once again divide, and they keep dividing in that same plane. So they're forming one long chain. When we get into bacteria that form the tetrads, you'll see that the cocci first divide in this plane, and then they're going to divide in the next plane up, so that two of the bacteria daughter cells will land up like on top of the two that are below it. So they make like a square shaped tetrad. And when we get into the sarcinae, they first divide, in the one plane, then they divide in the next plane, and then they divide in the Z plane going forward. So you end up with a cube. Staphylococci, they're going to divide just irregularly in kind of a willy-nilly pattern. And that's why they end up in these grape-like clusters as they just sort of bunch around. All right, so here you see the planes of division. Again, the diplococci just divide in half. And the streptococci they're just dividing over and over again in that same plane, okay? The tetrad, you see here, they first divide in the one plane, and then they divide in the plane next to it. Sarcinae divide in all three planes, and so you end up with this cube-like cluster. And staphylococci all over the place. But a lot of our bacteria, especially in the bacilli group, will probably exist as a single bacillus. You're not going to see, here we have a couple of diplococci, and they might actually later on separate. We might just have caught them as they're dividing and still stuck together, but you can see the vast majority of these are existing as single diplococci. Like this one looks like it's just getting a cleavage furrow in it right there, that it hasn't completed division. Same thing, coxobacillus, kind of hard to tell, is this bacilli that's just like dividing with the cleavage furrow in it and will become an individual cocci or bacillus. A lot of times they just kind of exist in this sort of intermittent or in between stage. Some features of a coccus rounded at the ends, but it's a little bit more elongated and oval shape. So you can legitimately just call it a coccobacillus if that's what you see under your microscope. Here we see that diplobacilli is just like our cocci that are diplococcus, and they divide in the same plane. And the same thing with streptococci. They just keep dividing in the same plane. Looks like a long string of hot dogs here. And then a more unusual, but sometimes we see it, are going to be some bacteria that form these palisades. And so they kind of bend and stick at this end, you know, and then at this end, and then at this end. So they're individual bacteria really, but they kind of ended up lining up like a picket fence right here, okay, so where they'll be stuck together on one end. All right, so we'll talk a little bit more about the bacillus. The bacillus, of course, are our rod shapes. And we see, how would we identify Bacillus anthracis here? Anybody? They're going end to end, right? So this would be as a streptobacillus, right? You see, strepto means forming chains, just like streptococcus will be one, you know, little rounded bacteria stuck together in a long chain like beads on a pearl necklace. Okay, so here we have our idealized structure of the prokaryotic cell that we've talked about before. And here you'll see all the parts that we're familiar with. We have the nucleoid, which is where the bacterial DNA is. And the bacterial DNA is not enclosed in a nuclear membrane as it is in eukaryotic cells. Remember, we talked about that, how there's a nuclear membrane in eukaryotic cells, which serves to create an extra barrier and protect and encase our DNA. And the bacteria don't have this. And the bacteria also have a higher mutation rate. They're more susceptible to things like ionizing radiation. The DNA of the bacteria is not wound around histones, but you'll see that they are associated with some nucleoid-associated proteins. Outside of the nuclear region, you'll see there's a plasmid here, and the plasmids are a lot like a virus, but they aren't a virus. And they are considered naked DNA. And, oops, excuse me, this trigger is really happy on this mouse that I have on the new iMac. And this naked DNA can move from one bacterial cell to another, especially through a reproductive pilus or a conjugated pilus in a process we call bacterial conjugation, which is bacterial sex in quotes. It really isn't a sexual mixing of genetic material, but simply a way that some DNA gets passed from one bacteria to another. And when this happens, the passing of a plasmid through the pilus, most often the plasmid can confer some selective advantages to the bacteria, such as the genes that are contained on there can be genes that might confer resistance to antibiotic or antimicrobial substances. These are not essential genes for the existence and life form of the bacteria that are on the plasmid. Those are all contained in the nucleoid material. As well, you'll see in the cytoplasm, there are free ribosomes. They're not bound to any sort of membranous organelles like the endoplasmic reticulum. Okay, the ribosomes in a eukaryote are 70S ribosomes, whereas in a eukaryote, like our cells, those are 80S ribosomes. So you can see that because the ribosomes are different, those two will be an antimicrobial target of some of our drugs when we get into that. All right, looking at the structures that are outside, we see first this yellow area, which is the plasmalemma, and the plasmalemma in a bacteria is much like ours. It's a phospholipid bilayer. Outside of the plasmalemma, you'll see we have the cell wall. And remember, the cell wall in the gram positive is thick with peptidoglycan, whereas in the gram-negative, it is thin with peptidoglycan, which is sandwiched between an inner and outer membranous layer. You'll also see outside of both of these is what's called the glycocalyx, or it either forms a capsule if the glycocalyx is organized, or a slime layer if it is a little bit more amorphous, okay? And that capsule or the slime layer can confer a big advantage for survival on these bacteria. And we call this, remember this term, a virulence factor. Why it's a virulence factor is because all that polysaccharide coating that's secreted around that bacteria can cover up a lot of the antigens from the lipoteichoic acid or from the lipopolysaccharides, and cover up the antigens that we typically our antibodies see. It helps basically the bacteria evade many of our immune cell processes. Projecting from the plasma lemma and out here, you'll see a pilus, and the pilus and the fimbriae are kind of related. The pilus, in the sense of for bacterial conjugation, is more of this hollow tube really than the fimbriae, but you'll see a lot of people classify them together, and I'm not going to get too picky about it, but you'll never see anything being called a reproductive fimbriae. It will always be called the conjugative pilus or reproductive pilus. Okay, these fimbriae also extend out. You see many of these going around here, and these fimbriae are also allow the bacteria to adhere to each other and to the host cells. So say like if you have a strep throat or something, those fimbriae can adhere to the mucosal lining of your throat, for instance. As well, we also see out here the flagella, okay? And you'll see the flagella is kind of lodged deep down into the plasma lemma, and the flagella has a little like motor that's operated on a proton electromagnetic force. There's a concentration gradient of protons, similar to what happens inside the mitochondrial membrane for making ATP. And that drives the energy of the motor to spin these flagella as kind of a rotatory motion. In gram-positive bacteria, there's sort of two parts of the basal body that anchor this sort of basal body anchoring system, whereas there's sort of four anchors to in the basal, in the gram-negative cells, because of the inner and outer membranes there. And this has a little hook region we'll see on another slide, and that extends down into the flagella. And the flagella are made up of a protein called flagellin. So don't confuse this with like the tubulin that you'll see in the eukaryotic cells, okay? All right, is there anything else on here I'm missing? Oh, these inclusion bodies. We'll go through the inclusion bodies at the end of the lecture, and you'll see these are things like the vacuoles and the magnetosomes and such that either can help the bacteria direct their movement or orient them in the level of water if they're aquatic bacteria. All right. All right, so we'll discuss each of these structures separately, and I have it written down so that you can remember it. So the glycocalyx is, again, that sugar coating on the outside of the cell wall, and it's viscous, it's gelatinous, and it's sticky. So it's made of all these polysaccharides that might also have some peptides in it. And as I said, two main types of organization: the capsule, which is a little bit more organized and maybe a little bit hardened and helps prevent the cell from desiccating, and a slime layer that's a little bit more amorphous, a little bit more disorganized or loose. And both of these help the bacteria evade the immune system, hence making it a virulence factor. But as well, it can also help the bacteria stick to each other, all that sticky substance. All right, so as I said, the bacteria is a virulence factor. And when we're talking about virulence factors, which we'll talk about a lot this semester, are going to be qualities that the bacteria have that will help them become a pathogen, okay? It helps them survive and evade our immune system. And the capsules, like I said, prevent phagocytosis by the host cells, things like your macrophages when the bacteria initially comes into your body. They also will help form biofilms. And this is because they secrete that sticky substance and will help the cells bind to each other, will help the cells bind to things like IV lines, help them adhere to a surface such as the biofilm, which is the plaque that forms on your teeth, okay? And here you see, remember, the negative staining of the capsule. So you see the little halo around the cell that does not stain. And again, here's another slide to sort of show you looking at the outside of a rod-shaped bacteria. This is more loosely organized. The slime layer, it's a little bit thinner, and the more organized, thicker bacterial capsule. And, oops. And here is the glycocalyx again, just to remind you that it can form those films, such as on your teeth. I used to work with a surgeon who had terrible plaque on his teeth, and it used to drive me insane. I used to want to tell him, please go to a dentist and clean your teeth. But you can see that, you know, that plaque forming thickly around there. And it's all that bacterial adhering to each other, but also adhering to the surface of the teeth. And having plaque is actually an interesting marker of your overall health. And they found that people who have a lot of plaque usually are at higher risk for things like endocarditis. And it's not sure if it's related to the actual bacteria that's in the plaque, because those are different bacteria like strep mutans that cause the cavities in the biofilm in your teeth, versus the kind of bugs that actually cause endocarditis. So they think it's just like a marker whether you take care of your teeth as to whether you take care of your overall health. But I'd need to look that up and see if there's any new research on any of that. All right, so here you can see from the interior surface of your teeth, some more of this plaque on your lower teeth, which is really one of the more common spots that plaque forms as your tongue thrusts a lot of what you've been eating and the sugars and that in your diet up against the teeth. And then it calcifies and hardens, and the plaque becomes harder to remove. Okay, talking about our flagella. The flagella are these filamentous appendages that extend from the cell wall, and they're made of kind of a polymer or fusion of the protein flagellin. And their function is to propel the bacteria forward. It's, you know, a motility function. And the flagella will spin in a rotary manner. And like I said, it's going to be a proton motive force that powers that. And this flagella will kind of spread out, and there is going to be like a hook in a basal body region that extend into the shaft of the flagella. I think we see that on the next slide. But remember that the bacteria don't have the motilin or dynein that you see in the typical flagella that we'll talk about in eukaryotic cells, or we've talked about in anatomy in the eukaryotic cells of like what the flagella and the sperm are made of. So we have three parts to our flagella: the filament, which is made of the protein flagellin, and that will get rotated out from the motor through the hook. And we'll see the hook is what attaches both to the filament, the long end, and the basal body that anchors the flagella into the body proper of the bacteria. The basal body is going to consist of a rod and two pairs of rings. Okay, so you'll have a pair ring in the gram positive and two pairs in the gram negative. And it anchors that flagellum into the cell wall and plasma membrane. And the motor of the flagella will end up kind of moving this. And we'll see this better in this picture here. Okay, so here we have a gram-negative. So this is a picture of a gram-negative bacteria. So you see the filament, that's the polymer of flagellin. And here's that hook region. And you can see, because we have both an outer and inner membrane here, that we have the 4 different rings, whereas you only have 2 on the gram-positive. So you see this, the L ring associated with lipopolysaccharides, the P ring associated with the peptidoglycan layers right down in here. Remember, the peptidoglycan runs here through the periplasm. And then you'll see the M ring that's embedded in the plasma membrane. And then this S ring is the innermost that's directly attached to the plasma membrane. Here we have different arrangements of the black bacterial flagella. You have the paratrichus, the monotrichus, the lophotrichus, and the amphitrichus. And it's easiest to see on the monotrichus, where you just have the one, how this flagella will spin around and it's going to launch the bacteria to move kind of in the direction at the opposite end of the flagella. If the flagella is moving and spinning out here, the bacteria is going to move in this direction away. On the lipotrichus, you have multiple flagella, but they're all at one end or one pole of the bacteria. So that too moves the bacteria in one direction. The amphitrichus flagella is a little bit more interesting because we have it, we have it at both ends of the cell. And so what you'll see is the bacteria will use one flagella to move in one direction, and then it'll stop that flagella and use the opposite flagella to work when it wants to go in another direction. Okay, and this paratrichus is a little bit more confusing. This is going to kind of has the bacteria being able to move in different directions, and we'll talk a little bit about bacterial runs and tumbles and get to that a little bit later when we understand the motion a little bit better. And then we're going to have an interesting new type of axial filament that's really in our spirochete bacteria. It's called an endoflagella. And this, you'll see, is kind of anchored at both poles of the spirochete, runs through it, and it's kind of wound around, and it's in the periplasm underneath the cell wall again. And so this will allow that kind of corkscrewing movement that the spirochetes make. So you can see right in here, anchored at both ends, and it kind of runs through, it's running through the periplasm, and it kind of just allows that to corkscrew and twist around that axial filament. Here's an up close picture of that axial filament kind of running through. Sorry, I'm really sorry about the way this mouse is. I got to get used to that. Fimbriae and pili often classified together. Just remember that for pili, that will have that special conjugation pili. You can have many of these, a few or hundreds, and they allow for attachment. If you want to split the hairs, get on this one, split the hairs of pili and fimbriae. Mostly we say the fimbriae are used for attachment, like to attach to cells or to help attach to other bacteria, and the pili more specifically for some movement and especially for conjugation, okay, transferring a plasmid. All right, so you can see all these projections here of various pili or fimbriae. You can see how it would be very difficult to tell a fimbriae apart from a pili. All right, and as I said, the pili typically thought to be a little bit longer than the fimbriae. These can be involved in some gliding and twitching motions, but the one that's important to remember, again, is the conjugation pili that will allow plasmids to transfer from one bacterial cell to another. The cell wall, of course, is the semi-rigid structure that gives the bacteria its shape. It's also what is stained with our important stains like the gram stain or the acid-fast stain. And so that's useful for us. And of course, we classify our gram stains: gram positive, thick peptidoglycan; gram negative, not as thick peptidoglycan, and has the outer and inner membrane surrounding it, sandwiching it. The cell wall will contribute a lot to the pathogenicity of the bacteria. You know, it allows the bacteria to be protected from things like lysis. It helps prevent desiccation of the bacteria. And you'll see that many of our antibiotics will frequently target the cell wall, particularly when bacteria are dividing and they need to synthesize a new cell wall. That's going to be a real good point where our antibiotics work. And such a bacteria would be penicillin that will stop your bacteria from forming these nice little binding points or links between the peptidoglycan so that holes are allowed to form in that cell wall. And then, of course, water, which is the universal solvent in our bodies, can then osmotically rush into the bacterial cell and lyse it, break it apart. Okay. And here you see the transmission electron microscopy picture, TEM, of a prokaryotic cell and the structures that we've been talking about. Remember that TEM, transmission electron microscopy, shows you that really up close submicrometer level of the interior of cells, whereas scanning electron microscopy, SEM, is something that displays the outer part of the cell. All right, so looking at our bacterial cell walls, we'll start this time with gram-negative cell walls. And you can see here that it has that thin peptidoglycan layer, which is made up of alternating units of NAG and NAM. NAG here being the red N-acetylglucosamine, and NAG being the N-acetylmuramic acid. Okay, and these polymers will form, you know, long structures that are then interlinked with some side chains of some amino acids that hold them together, but just a thin layer of it. Here in this periplasmic space, and there will be periplasm flowing around it. And then we have our inner membrane, which is just basically a plasma membrane with some integral membrane proteins in it. And you see your phospholipid bilayer. Where the action is here in the gram-negative is in this outer membrane, which is again a phospholipid bilayer, but in here inserted in, you see the lipopolysaccharide. And if we look up close at what the lipopolysaccharide looks like amongst the phospholipid bilayer, is you see it has a lipid A component, which is also the endotoxin component that is attached to a core polysaccharide and an O polysaccharide. And this O polysaccharide will be significant for antigenic properties of the gram-negative cell wall. Here in the gram-positive cell wall, we see we just have an inner plasma membrane, okay? Phospholipid bilayer with some integral membrane proteins. And then you have a thick peptidoglycan layer, which again is alternating units of NAG and NAM. And on a test, I won't make you have to come up with N-acetylglucosamine or N-acetylmuramic acid. If you just want to remember it as NAG and NAM, that's good enough for me, okay? So this forms that big cell wall layer. There's no outer membrane as there is in the gram-negative. All that thick layer after layer of peptidoglycan, and it's all covalently cross-linked again with the same side chain amino acids in both directions, linking up between the layers and across ways between the various NAG and NAM components. But what you'll see is largely different is there's going to be teichoic acids. Some of them will be lipoteichoic acids that will span from the phospholipid bilayer, and some will just sort of be in between the NAG and NAM layers, the wall teichoic acids. And then, whoops, sorry again. And we will have some that are granular in this granular layer, just sort of resting across through the periplasm. Now, these teichoic acids will also be antigenic in the gram-positive bacteria. All right, here we see the gram-positive structure up close again, or I should say the idea of the peptidoglycan up close. Again, NAG, NAM, NAG, NAM. And then here you see the cross-bridging amino acids for that horizontal cross-bridging. And then you see the vertical side chain amino acids, okay, that link in here on the N-acetylmuramic acid. So this is that carbohydrate backbone of those sugars of NAG and NAM. And then you can see the tetrapeptide side chains right here. All right, so peptidoglycan is also known as murine, more often in the older literature, more often today, most everybody refers to it as peptidoglycan, which it will always be regardless of whether it's gram positive or gram negative, these repeating units of the disaccharides of NAG and NAM. And they'll be linked by those polypeptides that we saw. And all bacteria are going to require having an intact cell wall to, contain its cytoplasm within and protect it from osmotic lysis from the different osmotic pressure on the outside of the bacteria. Okay, so that cell wall is an important component for that. And as you'll see, it's what we exploit, trying to find weaknesses in the cell wall, because the cell wall does not exist in our eukaryotic cells. It will be a target for when we develop any antimicrobial drugs. Gram positive, as I said, have thick peptidoglycan layers that contain the teichoic acids, okay? And just like the O in the outer, the O saccharide in the gram negative, because it juts out, it's going to be a part that is going to have antigenic specificity. Okay, we can detect what bug we have by what teichoic acid is linked to it. And our teichoic acids will have a negative charge that is both gram positive and gram negative bacteria have negative cell walls. Peptidoglycan has a negative charge on it, but also the teichoic acids of gram positive will have negative charges on it. And this will bind to and regulate the movement of cations into and out of the bacterial cell walls. Underneath, in between the cell wall and the plasma lemma, we're going to have what's called the periplasmic space. Okay, and so this is going to be containing a periplasm, and it will also contain within it that granular layer of the lipoteichoic acid, which is also similarly negatively charged. They're actually using positively charged nanoparticles as a way to try to attract the negative charges on bacteria in some filtration systems today. Okay, so the teichoic acids will link to the cell wall to the plasma membrane, and the wall teichoic acids will link to the peptidoglycan. And remember, they all have negative charges on them, so they regulate the movement of cations into or out of the bacterial cells. All of these, the polysaccharides in those teichoic acids will again have antigenic specificity, meaning our antibodies can be made against those, point of attack, right? The negative gram negative cell walls contain thin layers of peptidoglycan. Here, the outer membrane contains lipopolysaccharides, all right? Lipopolysaccharides. These also have negative charges that help the bacteria evade phagocytosis by the host immune system, all right? Running in between the inner and outer membranes, you'll see where that teichoic, where the peptidoglycan lies in the gram negative cell is within the periplasmic space. And it contains, again, just like in the gram positive, periplasm, which is a gel-like fluid in that periplasmic space, much like cytoplasm inside the cell. Different on the outside of our gram negative cells is the lipopolysaccharide component. And the lipopolysaccharide component blown up looks like this, where we have our lipid A portion, also known as endotoxin, okay? We'll talk a lot about the toxic effects of endotoxin and how it leads to endotoxic shock. And endotoxic shock will cause things like your blood pressure going through the floor, your temperature going up, really labile state, okay, a septic shock. And the lipid A component is what is responsible for this. Attached to the lipid A is the core polysaccharide and also the O antigen or O polysaccharide. This whole unit together is the lipopolysaccharide. So lipid A is the endotoxin causing, that massive fever, shock, blood clotting, blood pressure going through the floor. The core polysaccharide attaches to the lipid A, all right? And the O polysaccharide extends out from the core. That O polysaccharide extending out from the core will function, like I said, as an antigen. So when we see things like E. coli O157H7, this O is referring to that O polysaccharide. And this E. coli is an enterohemorrhagic. It's the terrible E. coli that people got, hemorrhagic diarrhea, literally bleeding out from their rectums when they ate undercooked hamburger, especially at, it was notorious at the Jack in the Box chain of restaurants, which maybe many of you might not be familiar with. Jack in the Box was around like McDonald's and Burger King, but you don't see it anywhere anymore. And it's largely because they had a terrible, scandalous event of a lot of bacterial hamburger being hamburger with bacteria of E. coli O157 type sent out across the nation, and many people died. All right, and then you'll see that we have a lot of porins in our cell walls, and these porins are proteins that form membrane channels, much like a pore, giving them their name, a porin. And this will be forming these channels that where the uptake of small molecules will occur through that outer membrane in the gram-negative bacteria. All right, so once again, you see your gram-negative and your gram-positive. Here we see in the gram-positive our thick layer of peptidoglycan, the periplasmic space containing the periplasm, and the phospholipid bilayer with the integral, I can't talk anymore, integral membrane protein, okay? Out over here, you see the gram-negative with its thin layer of peptidoglycan in the periplasm, which is located in the periplasmic space. You see the inner membrane of the phospholipid bilayer with an integral membrane protein, just like a normal, plasma lemma. But it also is encased in this outer membrane. And this is where all the action is in the gram-negative bacteria, because you have the pores that can let some cations and stuff in, or nutrients in, and also let some substances flow out. But we'll also have our phospholipid bilayer of the outer membrane, from which extends our lipopolysaccharide with its lipid A, coarse polysaccharide, and antigenic O polysaccharide component. Now, as we have learned, our cell walls will gram stain, and both our gram-negative and our gram-positive will initially stain with the crystal violet iodine stain. But when we use our acetone alcohol to wash away the stain, it only washes it out of that thin layer of the gram-negative bacteria. It is retained in the gram-positive, and the mordant of iodine, right, is what helps that crystal violet stay crystallized and dyed and bound to the gram-positive layer of peptidoglycan. The gram-negative then, at the decolorizing with the alcohol and acetone stage, will lose all that dye. And so after we decolorize, the gram-positive remains purple, but the gram-negative is non-colored. It only colors, again, when we add the counter stain. such as safranin, okay, that will stain it pink. And here we see gram-positive bacteria stained purple and gram-negative bacteria stained pink. As well, we have some atypical cell walls. And the atypical cell wall of the mycoplasma is a really thin layer. Well, let's go back. The mycoplasma are our smallest bacteria, and they actually lack a cell wall, okay? They have no cell walls or very, very little cell wall material. So gram staining is ineffective for them because they require, they don't have a cell wall really to stain. Even that very little bit of cell wall will decolorize in gram staining. So we need some special staining techniques for that I don't think we're really going to talk about staining of mycoplasma in this course, all right? But you use some DNA kind of fancy technologies with fluorochromes and something called a host HOEST stain. Now, lacking a cell wall or having a very minimal cell wall means that, you know, the cell wall is what gives the bacteria its shape. And so they don't have a very consistent shape. So these are the bacteria that end up being pleomorphic, taking on many different shapes. So sometimes they'll be a little comma shape, sometimes they'll be a little fat like a round little cocci, and other times they'll look more rod-like, all right? These, again, are the smallest bacteria with the smallest genomes. And they're not easily then destroyed, obviously, like by any of the antibiotics that we have that attack the cell wall, because they don't really have a cell wall. They're so small, so tiny that they actually pass through most of our bacterial filters, and they're notorious for contaminating labs. And so in really highly specific labs, you have to do a lot of things to protect against invasion of mycoplasma. Their plasma membranes are unique in that they contain a lot of sterols. So not in the cell walls. We're talking now, even though it says atypical cell walls, we're moving down into the plasma membrane that contains sterols. Okay, sterols are like cholesterol, right? We all have sterols in our cell wall, but they have large quantities of the sterols in their cell wall. And these sterols kind of give them a little bit of fluidity to the membrane, just like cholesterol does to ours. And an example of mycoplasma is going to be Mycoplasma pneumoniae that gives you walking pneumonia, which is an atypical sort of pneumonia. And it can be very difficult to treat, very difficult to eradicate. And a lot of people will walk around with this for a while, hence the name walking pneumonia, and they just think they got a cold that's hanging on. And then one day they get, you know, really profoundly ill. These are different than mycobacterium. That's why I have mycoplasma do not equal mycobacterium, and your brain is going to want to confuse the two. So just remember, mycoplasma, pleomorphic, not really a cell wall. Mycobacteria have the acid-fast cell wall, okay? These are slender, curve-shaped rods. They have a thick, waxy cell wall around the cell membranes, and it has three major components to the cell wall. Peptidoglycan as another bacteria. They also have a thick lipopolysaccharide layer, kind of like gram negatives. And they're going to have an outer membrane that contains a lot of this waxy substance called mycolic acid. So the example of this is going to be Mycobacterium tuberculosis, which causes tuberculosis, TB. Okay, and remember, these are what's going to stain acid fast. We went through acid fast during last lab. But we'll keep going through this stuff so it stays in your mind. So in acid-fast staining, as you recall from lab, we have two methods, the Ziehl-Neelsen method and the Kinyoun, Ziehl-Neelsen using heat to help imbue that cell wall with the carbofusion dye that we're going to use. Quinine doesn't use heat. It uses a higher concentration of the stains for longer periods of time. So Mycobacterium, like I said, is going to be Mycobacterium tuberculosis, but also Mycobacterium leprae, which causes leprosy. And there's another species of bacteria that's also acid-fast called the nocardia. And these are some gram-positive acid-fast rods, and they form long filamentous chains. And there's only like one strain that's really medically important, and we're really probably not going to get into nocardia in here. Okay, we can't cover all the world of bacteria. All right, so when you acid-fast stain, remember you use carbofusion red, which is much more soluble in that waxy mycolic acid. It's lipid soluble, and it contains phenol, a form of alcohol that's off of a benzene ring. And this helps the stain to penetrate along with the application of heat. So then we'll use very strong decolorizer that will remove the carbofusion stain from any non-acid fast bacteria, but it's stuck there in that waxy substance of the acid fast cells. So then the decolorized bacteria will take up whatever counter stain you're using. All right. Now let's talk about structures that are internal to the cell wall, beginning with the plasma membrane. Again, our plasma membrane is a phospholipid bilayer that encloses the cytoplasm, okay? And that's how we compartmentalize from the outside world. You have the hydrophilic heads facing each side, either, in a case of the eukaryotic cell, facing the outer environment, or the internal cytoplasm are the hydrophilic heads. So it's facing whatever's watery. We have our hydrophobic head and our fatty acid tails that are hydrophobic in our hydrophilic head. And we have two of those, and they face each other. So we have all of our hydrophobic tails intermingling together, having basically essentially two layers sandwiched in. They're hydrophobic that will repel anything that's polar, repel all that water, okay? But we have our hydrophilic heads up here where my thumbs are curved in, which are facing the aqueous environment, all right? All right, and embedded through all that, we'll have a lot of peripheral membranes, peripheral proteins in the membrane surface, and we'll also have some integral or transmembrane proteins that will span across the entire range of the phospholipid bilayer. And a lot of these will form some of the channels that will help let substances in or out of our plasma lemma. All right, so here we have a diagram of the lipid bilayer that forms the inner membrane of the gram-negative Vibrio cholerae, okay? So a curved, here you see the curved, a little bit comma-shaped Vibrio cholerae, and you can see our phospholipid bilayer here interior to the cell wall on the outside, okay?
I have a question. It's a little off topic. Just to clarify, what time does class go till tonight?
Isn't it supposedly 7:55? Are we beyond that already?
I thought it was 5:30 to 6:45. I just have to work, so I didn't want to miss too much of lecture. I was trying to hang on as long as I could.
Oh, okay.
I could be wrong. I thought that's what Tuesday was.
All right.
I think it goes till 6:55 too, is what our schedule says.
Yeah, that's what it says.
Let me double check.
I just didn't want to miss too much, so I just wanted to
No, I
clarify.
know. We started at 6 tonight, so I'm trying to share with you the schedule. Can you guys see the schedule now? Whoops.
Yeah.
Oh, it clicked into the thing here. So sorry, it clicked into the Washna. Yeah. So yes, 5:30 to 6:55. So we started late. Sorry about that. All right. So somebody, remind me when class ends, because, I can't see very well and I have to periodically check periodically check to make sure that we have, that we're not going too much overtime. So we're at 7:15. I guess we should end and pick up where we left off then. Is that good for you guys, that we'll pick up with the plasma membrane next time? Does that sound okay?
Yeah, that's fine. Thank
All right.
you.
Okay. Did nobody get my message about class starting at 6 tonight?
I
Oh.
did not.
No,
And
I didn't. Oh, I didn't.
There was some technical difficulties. A lot of people could not log on. And so I had
Yeah,
emailed
so
you.
that's why I sent out a message. I didn't know if the message was going through either. There were a lot of technical difficulties with Zoom for some reason. It was not letting anybody sign on, me either. And so,
I got a message from WCC2 right after we started class, and they said Blackboard was down for a
I
while.
got that too. That's what I thought was going
okay,
on.
so that must have been what the problem was. And that's why through Blackboard, I tried to send you all a message saying that, we'll probably start at 6 because there were all these technical problems. So I apologize if that message didn't get through to you. And I posted it on the announcement section. But again, that might not have been an adjustment that was made because of all the technical problems. I hope my posting on Blackboard and that the slides, I guess some of you had trouble getting the slides from Blackboard.
They popped up at like
Yesterday.
6:10.
Okay, so it did pop up at 6.
Well, the
So maybe
problem
they got
was,
rid of all their technical problems at that point.
and I apologize, I emailed you. This is Danielle. I emailed you a couple times because Blackboard wouldn't even pop up on my computer. I couldn't even get on to be able to see an announcement or anything like that. So I was like, I apologize, but you
Yeah,
can just.
no, that's okay. And
I
this,
had that
this is-
issue too.
Yeah,
I called your house and,
Oh, okay. This is stuff that we can't really, none of us can do anything about the technical glitches of working online. This is something we had to deal with last semester and not deal with it too much. Last semester, I had problems with my WoW internet, which, like, cross my fingers, is going to hold out this time. And we're just going to have to do the best we can to be online. And if ever you want, to go overtime, like I said, somebody stopped me when I'm going overtime. I don't know if I told you that last semester, it was the end of winter semester. I had a healthcare attendant here who was taking care of my mom while I teach, and she stole my watches. So the watches that I had keep laid out here on my desk so that I can keep track of time while I'm lecturing to you. Because when I turn on the PowerPoint slides, the little clock goes off on my computer and I can't see if I go overtime. So don't be scared to pipe in and say that we've gone overtime and we got to stop. All right. And we'll just pick up where we left off. So we'll pick up with the plasma membrane next time. If we're not really getting done with prokaryotes by the end of next lecture, then we won't start the PBL. I have it right now set that we'll start on September 17th. Then we'll go over and not start it until after lecture next Monday. All right. So we just got to see where we can get, since we lost a half an hour of time from all that rigmarole at the beginning of the class today. All right.
Okay, and what are the PBLs like? Like, are they kind of like our homework where you can open them and close them and then open them again, or do you have to do them all in one sitting?
Yeah, no, you don't have to do them all in one sitting. I usually give you like 3 or 4 days. They tend to be flooded. I actually have fewer questions for microbiology than I do for anatomy and physiology, because anatomy and physiology, you have an endless amount of material and questions. Microbiology, I would say it might be like 100 questions. They're not, because we're still online, they're not true PBLs. Like if I were doing a PBL in class, I would probably give you more of a deeper clinical scenario and have you think through clinically how you can apply these what you're learning in microbiology, because ultimately everybody in our class really wants to learn to think clinically about the bugs and drugs that kill them. So you will have some questions that'll be just like a regular test question of fact-based things that you have to know, like, here's Staphylococcus, Streptococcus, E. coli, Vibrio cholerae, which one stains gram negative, you know, and you'd pick out your E. coli. So some fact-based stuff like that, fact-based things about, you know, what component of lipopolysaccharide is considered the endotoxin, and you'd pick the lipid A, things like that. And then I'll have other things where like I might have you read something about a bacteria and you can relate things to it. Like I'll give you a lot of information about, and sometimes I make up bacteria names, and you can tell me what you might think would be the virulence factors for this bacteria. If I tell you, you know, that it stains negative with a negative stain, and it stains pink on a gram stain, and it has a flagella on it. And I'll say, you know, list 3 things that are virulence factors. And you could pick like the capsule, the flagella, you know, and like lipopolysaccharide, you know, because it was gram negative, something like that. Okay. So it's kind of like a 2-step that's a little bit more problem based. I know I have a couple of bigger problems. A lot of the students don't like those that make them think a little bit more, but this is what tends to separate the A students from the B students is, can you really think through a clinical scenario? I know last year, and I'll probably give it again, I gave a question with a clinical scenario about a specific bacteria, and I had people apply Koch's postulates to it. So you had to tell me from the stage of discovery of this bacteria where the research was in terms of proving Koch's postulates. You know, go all the way through and prove, you know, that the bacteria caused the disease, or was he at the stage where the bacteria just showed that it killed, it was, you know, inside all the dead animals? Had he yet given the bacteria to, you know, live animals to kill them with it, you know, something like that. So they're not too hard. I literally gave half the class, about half the class got a B+ or better last time. The other half class was mostly separated between B, B-, and C+. I think I had to give one person a C-, which I hated doing, but the thing that's got her to get the C- was that she didn't do a lot of the work. You know, if you just don't hand in the homework, I can't give you a grade, and there's nothing I can really do about that. So, all right. So you guys, I expect that you'll do really well. Just pay attention and I'll lead you. I mean, are you guys finding the quizzes hard?
No, I think
Well, they're intermediate.
Yeah.
Intermediate. They're very much
Yeah.
like the questions that I give you on kind of the
Some of
quizzes.
the questions,
And
require
they're
more-
open book.
yeah.
So was that your question, Kathleen?
No, I was just gonna say, yes, some of the questions I realize I have to apply more than just like a quick answer. So they
Yeah,
get me
yeah.
thinking.
You know, because anybody can look up an answer. So when we devise test questions, there are questions that we call like level A questions that are really just fact-based. Can you regurgitate or do you know information? Which is super easy on an open book test, right? Nobody should get those wrong. It's just simply, you know, yep, that's a fact. You can Google it when you're answering the question. Then you go all the way up to like type D questions, which are, you know, can you apply this question? Type C is more comprehension. Okay, so maybe you have to do a little bit more reading and like, did you comprehend some fine point, like really knowing the difference between like maybe lipoteichoic acid in gram positives is what makes it antigenic versus the lipopolysaccharide in the gram negatives is what will make it antigenic, okay, in the gram negative. So that would be like a comprehension type question. And the type B are kind of a little bit in between. Maybe it's like you needed to know 2 facts instead of 1 fact, okay? Kind of a 2-stage question where I might say something, show you a picture and it's got a mixed thing of, something stained purple and something stained pink. And I tell you it's a gram stain and you need to pick which one is the gram positive. So you kind of had to do a two-step thinking. You had to remember that gram positive stained purple, and you had to remember, like which organism it is on there that is staining purple. Okay. So hopefully the practice questions, and sometimes I actually take questions that are from your quizzes and your homeworks and I put them on the test. So you have a second stab at some things that maybe you got wrong. And I know I need to go through, but it was due tonight because we had all the technical glitches and stuff. Early in the semester, we always have technical glitches. There's always problems with Blackboard, and then it gets better throughout the semester. So what I'll do is, tonight I'll go through and I'll hand grade all those things where you had fill in the blanks. And so your score should get updated by tomorrow. And so we should be all caught up. But our next set of homework and all that, I don't have anything where I have to hand grade anything. So you should be able to start seeing, where you're kind of parsing out and whether you need to do a little bit more or whether you're like right on track for where you want to be with getting your grade. I'm somebody who really believes in like nurturing my students and trying to help you along. And I am not looking to weed people out or fail people. I really want you to do well. Okay. And if, you believe you can do well, and I'm going to believe you can do well. And hopefully, in the end, it all works out. And if you're struggling and you have, some difficulty, let me know. And we can do some Zoom sessions individually, and we can try to, figure out what you're having difficulty with and where you're going awry and try to resolve the problems. Why I like to put some of the fill in the blanks and stuff is it makes me go through all your homeworks and quizzes individually. And I kind of get a feel for where people are having trouble and what you're having troubles thinking through. So I might throw in some projects where you have more things to like fill out, like in a PDF, those of you who had anatomy with me, or those you had anatomy with a lot of the other people, and they use PDF forms where you have little blue boxes that you can fill in answers and type out original answers, a little bit more essay style. And that also helps me see, if the class is struggling with certain things. So anything else? Okay. And I'm never afraid to go overtime. I sleep very little. I only sleep about 4 hours a night. So, if we need to go through midnight and if you want review sessions or anything like that, we can arrange to do them. I'm here to help you. Okay. Don't ever be scared of that. Don't be scared of calling me. Just please call me between the hours of 9 and 9 because I have a little 95-year-old lady who likes to sleep. Okay. Anything else?
What's due tonight?
What is due tonight? So by tonight, before class, you should have handed in all of your homework, I think, from, let me see, let me bring up the schedule thing again. I think it was assignments 1 and 2, right? And your lab from last week is not due until before lab the following week, right?
Okay.
I'm typically trying to make lab due on Wednesdays and giving you about 2 or 3 days to go through. Let me just bring up Blackboard so we can see it. Sorry, got to type in to get into Blackboard. All right, here, let's share it with you so we can see all the due dates. All right. Can you guys see Blackboard?
Yes.
All right. So see, I try to put everything in announcements here too. So Monday, you'll see this is what I put in before class of where I think we're going to be going on this week and what's due. So we're not going to do a homework this week. I'm sorry. We'll have a homework and a lab this week, but we won't have a homework after prokaryotes finishing the lecture next Thursday, because we're getting into the PBL and I don't want you having too much crazy stuff trying to remember it. All right, so this is the schedule, and I'm aiming now for the PBL to start about 5 o'clock on Friday night and go through, I think 9/22 is like next Tuesday, something like that. So you got about 5 days, all right? What has been due is we started out with assignment 1, 2 being due, but people had some problems with it. So I extended it to today. I think it was the last day that you could hand that in. And then I had a microscope being due yesterday, all right? Then we had biomolecules homework is going to be due, was due Wednesday, 9/15 last week, but I think we extended that also to be due tonight along with assignment 1 and 2 because of the problems people had with Blackboard.
9:15 is tomorrow.
Oh, 9/15. Thank you. I'm starting to lose track of my days. So all of this, I think, was being extended to tomorrow. Then we had assignment 2, lab assignment 2, also being due. This is what we extended this microscope one along with the staining one to be due last night. But I might have put it into being later on. Did somebody ask me to extend it till tomorrow? Anyway, has everybody handed in 1 and 2 of both lab and homeworks?
Yes.
Okay. All right.
Yes.
Late, because then assignment
Yes.
1 and 2 was due on
Yes.
the 11th.
Yeah, we had it due, but then
And
people wrote to me and said they had problems with Blackboard. So I kept extending it. Okay. So, you know, I've extended it a couple of different times because of issues everybody, you know, has with Blackboard in the class. And then I'm hoping, you know, today you're going to be assigned prokaryotes, and I have it for being due Friday, so that by the time I open up the PBL for you guys to be able to do, you should have been done with your homework. Okay. And I know prokaryotes is going into the next lecture, so that, so I didn't reassign a new homework for the next lecture. And then on Thursday, we're going to do a lab on aseptic technique in culturing streak plates. And so that is going to be due the day before we do.
under the sheet.
So you can always check in here to see when something is due. And then periodically, somebody in your class might write to me and say, Blackboard cut me off in the middle of while I was doing my homework and I had to go to work. And so can you extend, you know, the due date till like tomorrow morning so I can come home and do it when I come home from my night shift at work. And so I'll just change the due date for everybody, you know, in Blackboard. And so you might see a little variation, like when you go in here to, let's see, say like if I go into Blackboard and I do lab, I go on to lab and I look at microscopy and staining. And I go in here where I put in the due dates. Is usually way down here at the bottom. I put in when you display it till, and then there's a box for me to fill in the due date, which has passed on this one. So, okay. So I might adjust it on there on occasion. Now, if you want to see a PBL, what they're like, I'll go to one that comes like at the end of the semester or something that comes up. I'm not going to show you the one now, but so you begin the PBL and it just goes through here. It tells you how to do it, you know, and please keep your, you know, try not to do it on a cell phone or something, but it just goes through, you know, a bunch of questions. So here you have 128 questions on this second PBL and you move one after the other through the questions. When you take the PBL, you can save the answer, okay? And then you can close out and you can come back to it. It's not until you get to the very end and when you're at the last question that if you answer it, it'll have save and submit. Don't hit save and submit until you want it to come to me. You know, only hit save and submit when you're done and you've checked through your answers. But otherwise you can save the answer, close out and come back and your questions will be answered. All right. So that's kind of what your PBLs look like. You can go through, you see, I got tons and tons of questions. Some of them will have diagrams, some of them don't. I wish I could see if there's one that's got a diagram. You see, we have a lot of immunology coming up. A lot of it's kind of the same stuff that you did in your regular biology. Over here, I have more, you know, chemistry that'll link into, you know, chemistry that I reviewed if you were having problems with those macromolecules. Down here, there's Ann Hayes, who's the chair of the department. When she teaches micro, there's some videos of her doing it. And maybe she does something different from me that you might find helpful if you want to take a look at some of her lecture recordings. But she also has practice tests here. And if you go on these practice tests, you can get in here and say, here's some winter 2021 exam items. You can go into her exams and do some practice questions. So she has a bunch of these in here, like extraordinary amount of questions. So that you might find helpful, okay? So I don't know if you looked at this. Another thing I wanted to point out is when people ask me for letters of recommendation, like this last week, I had to write 5 letters of recommendations from students I've had in the past semesters. This gives you really good advice, not just for asking me, but for asking somebody else. So that how to ask and what you're looking for is like basically give a faculty member at least a month, give them your CV and tell them things about you and what your goals are. So that when I write a letter about you, I can write a really nice letter that says a lot of really good things about a person, because I don't want to write a mediocre letter. They really want it to sound like that I know you. And I can show you like a typical letter from a super excellent student I had years ago. Okay, let me try to share this with you of like the kinds of things that you want to say. Okay, are you seeing the letter right here?
No, it's just
No?
the Blackboard page.
Just a Blackboard page. All right, let me try to get in. Okay, here's the letter. Now you see it? I don't think
Yes.
she would be embarrassed because I can't say nicer things like, you know, I want to write in here that she's an exceptional student. And I write in things like she did not miss a single answer to any question on any exam, any quiz, any extra credit assignment. I mean, that's an extraordinary thing to be able to write about somebody. And so, you know, she was writing to a PA program, which she got into, and she's now out working as a PA in a cardiology practice in Dearborn. And I like to put in things if we go down the road of like what your community service is. And, you know, if you've had any clinical exposure, like did you work as a nursing assistant? Were you even just sitting at the front desk, you know? And I want to be able to write about your ethics and character, because this is something the nursing, PA, nurse practitioner, and physician programs want. And then I can write down something like that. You know, I want to give you my highest recommendation. I am so pleased to write this about you. And I can write things about, you know, things we've talked about. Like up in here, I wrote about like how she really wanted to debate between whether she wanted to go to medical school or PA school, but she really liked the idea that the PA program allowed her to do, you know, was more focused on the primary care, which is what she was interested in doing. She wanted to establish those long-term relationships with her patients. And so when you can write a really good letter and write something that sounds personal, the program is going to say, hey, that student really made an effort to get to know the instructor. The instructor took an interest in this student because the student was so fabulous. Look at all the great shit she was able to write about this student. And it sets you apart. And that's what you want. So you need to give somebody time to write the letter, because whoever's writing the letter is also teaching classes and they have a life. And, you know, it takes me probably about 4 or 5 hours, because usually you also don't want the students to see your letters, but I always let you see what I'm writing about you so that you know what I'm writing, even if, you know, you've waived your right to being able to see your letters. So I just want to put that in there so that you know, and give your people time to write something about you and tell them the kind of personal information that you'd like in there. Also, if you had an academic problem in the past, like a letter I just wrote for somebody, he had problems where when he started out, when he was 18 years old, he was immature and he flunked out of school. And then when he came back to school, he was 30 years old, approached it with maturity, got A's and B's. And having me say the kid realized he was immature when he started school, he made a lot of mistakes, but then he came back, and I can write that shows fortitude. You know, it shows that he's highly capable, but, you know, and he grew up. And when I say it, it sounds like, you know, these are, you know, great things. I can make it tailor in a way that it sounds very positive about you. Whereas sometimes when the student writes that about themselves, it sounds like they're making an excuse. So, you know, I've had people in the past that maybe had problems with like an addiction or something, and I can write how you overcame the addiction and have now been sober for, you know, a decade. And, you know, it doesn't come across bad when I'm writing it about you, but it might not sound so good when you write it about yourself. Do you see what I mean? And these are the types of things your letters of recommendation can do. And, you know, your professors will really work with you on this, or they should if they're doing their job. So if a professor should also be able to tell you whether they can write a good letter or if they don't have the time to write you a letter, because what you really don't want is a mediocre letter. Okay, nobody wants a mediocre letter sent about them, because there's going to be a pile of students applying that have great letters. And oftentimes these can be the defining things, because if you got 10 people who all scored the same score on your entrance exam, and you got some people with mediocre letters and some people with great letters, they're going to go with all the people who got great letters. Okay, it's just simply the way stuff works. Okay. So sometimes students only give me a day or two, and that usually doesn't work well. But however, like for an employment letter, that's different than applying to a program. I can just say, you know, like I had a past student back from 2016 asking me for a letter of recommendation for a nursing job. And she was a great student, and I can write great things about her. She doesn't want her current employer writing her letter because she doesn't want them to know that she's thinking of leaving. So I keep all my records. I know who you all are. I'll remember you 10 years from now. I remember students that I had 20 years ago. I have a memory like an elephant and hips to match. Okay. So anything else about anything with class and what's going on? All right.
I'm good.
Okay. So we'll sign off for now, and we'll pick up on plasma membrane on Thursday and do our lab. Okay.
All right, thank you so much.
Yep. See ya.